Tutorial for a phylogenetic placement pipeline, example pipeline on insectphilo.org. Go to Tools and Pipeline 1. Phylogenetic placement of queried DNA barcodes onto reference phylogeny. So this pipeline will be run locally on your own computer in a Linux environment, and several software need to be installed before this is run. I won't for the moment cover installing these, maybe separately, but for now these need to be installed. And all we need to do is copy these into the shell to run these commands. So this is a integration of query data with references, and references contain, let's say, a phylogenetic integration. So first we need queries. These are typically, say, community DNA, for example, some molecular ecology study. So for the example, we will use data from Beth China, which is the world's largest tree diversity experiment, which you can see here. There's two sites. This is located in Jiangxi province, East China, subtropical, and a very large area. Each of these plots is about 700 square meters, and several hundred plots each have been planted a specific species distribution. So there are either no tree species, a monoculture, all the way up to 24 tree species. And we're trying to understand how this is going to affect higher trophic groups, and particularly valuable for DNA barcoding is Lepidoptera, because the caterpillars, which are predominant herbivores, which are most directly affected by tree species diversity, and also caterpillars being rather more difficult to identify. However, if you can match them to references, moths and butterflies obviously having very comprehensive reference data and very easy to identify. So this is a particularly valuable case study. So we're trying to understand what's happening to the Lepidoptera communities. These have been barcoded over several years, and this is the data we will download. So this was first set of this data was published a few years ago now, and it's available on NCBI, and we can access with the eSearch tool. eSearch searching the nucleotide database, and these are the set of accessions for this query set, and because we know the specific accession range, so we can use this format with the colon between the upper and lower. So the first command, eSearch, just gives information on what's available, and then we pipe this into the efetch command, which actually gets the sequences themselves in faster format, and pipe this with the forward arrow into this file, which we have named. So we have a file named bethleps.fas. Open the text editor. You can see typical NCBI format. These are called description lines for NCBI in the each FASTA entry, rather complex. Accession, species names if present. We've added some code. This can be used particularly in this data set, also the marker name. Now note, some of them have species names, some of them do not. Some of them at the family level, some of them very vaguely labeled, some of them at genus level, so varying ranks of identification. This is fine. For this purpose, we're doing integration in a phylogeny. This gives us phylogeny-based indices, so it is independent of the taxonomic label given to the query data. So for query data, you may not even have any taxonomic information on that, and that's perfectly fine. Back to the pipeline, so there should be about 1,600 sequences in this example query set. The ID lines are rather complex. Here's a simple bash command which will rearrange the ID lines. You can now see they're a bit simpler, just taxonomic name and accession. We need to keep the accession because there will be duplication in the taxonomic assignments. So various things we might do at this stage for this type of data. OTU clustering is one, taxonomic assignment is another. Now we don't really need to do taxonomic assignment for our current purpose, which is placement into a phylogeny. We should do some kind of clustering because for community DNA there's often identical sequences, because obviously there will be some species being represented many times. And if we build a phylogeny, a lot of implementations for a phylogeny assume a certain structure in sequence variation, and they don't like it if there's identical sequences. So we'll just do hierarchical clustering and remove identical or near identical sequences. We can use blastclust. You can obtain this from NCBI FTP site. This is unfortunately this particular software is not being updated at the moment, but it nonetheless is a robust and well written tool and is very permissive to the input data structure. So copy the blastclust command, clustering 1,600 queries at 99% sequence similarity using hierarchical clustering -L. I think this is the length of the pair alignment, and we're specifying DNA here. Let's look at our output file for blastclust, and you can see each OTU, we've just got OTU, is arranged one per line, so we have just over 500 OTU, and some OTU have a lot of specimens or barcodes, and some of them just have a single one. So this is much more efficient data if we can just take one representative per OTU. Much more efficient to analyze this data, but importantly we don't totally disregard other members. This is because these are obviously many of these are other representatives of a species and present, say, elsewhere in the plots, and we still need to integrate these into the analysis later. But for now we'll just take a single sequence for each of these OTU. And we use a Perl script for this. This is obtainable from GitHub. So I put a new repository, insect philo tools, on GitHub, and any Perl scripts associated with the insect philo pipeline I'll put onto here. So maybe you can, if you have a GitHub account, you can follow these. So copy the command in, and I'm just going to add path where I happen to have this script. And this simply, for each OTU, it takes a single representative FASTA format sequence for each, which you can see here. So now we have down from 1,600 sequences, we now have 1,500. Okay, so that's the plot data, that's the query data, community DNA, and we're going to profile this. So now we need something to profile against. Now, these are caterpillar herbivores, so we can profile against the phylogeny of Lepidoptera. And here's a very large one. Go to Lepidoptera version 2, which is over about 11,000 species. Right click, well, just click download here. That will go into your downloads folder, and so we have a Newick machine readable reference phylogeny, and we have a barcode dataset exactly corresponding to the terminals of that phylogeny. So download that also. And move these two files into your working folder. Take a look at the FASTA file. We have FASTA format sequences, and actually you can see right at the top is a single out group. These are aligned and 11,000. And we have the phylogeny in Newick format. So we need to now align the queries to the references. Now, because we have a nice neat alignment already, we can do what's called profile alignment instead of some de novo alignment. So profile alignment, there's various software, and I have put Clustral Omega in this case, which should work fine. Again, copy this into the shell and run. So Clustral Omega, we have the profile or the references, which we just downloaded from the website, and we have the query data. We specify an output, file name, and best to specify 2 iterations for the, I think this is the guide tree, and because if just doing the default, then you can get a few edge errors in the query alignment. Always best to check alignments by eye. BioEdit is a Windows software which is very good for this. For Linux, you might want to try Eugene, which has a user interface. And you can see this is perfectly aligned. So note the queries. Incidentally, we have renamed, or we've added a string to the queries OTU to the start. This makes it easier to find them later. But the queries are in the first part of the file, then we have all the references in the remainder. But note they're all in the same file. Some software for profile alignment will only include the queries in the output file. So the last step is phylogenetic placement. So let's look at the commands. It's actually split into 2. This could be done in one step, but in practice this seems to usually this will crash after doing the first step. I find it preferable to do like this. So dash capital F, skipping the final optimization, just getting the topology. We have the reference tree. We have the alignment of references and queries, output file name, DNA model, of which there is only one, and single outgroup. Running at 2 steps, and the second step is just doing model and branch length optimization, hence using the fixed topology of both references and queries. So copy both of these into the shell. Wait about 3 hours required. This is a large data set, remember 11,000 just in the references, and it will take about 1 GB of memory. But when this is done, we will have a large phylogeny of both the references and the queries, which we can view with a tree viewer, and I recommend Archaeopteryx. So remember, we added the string OTU to the faster IDs when we had a step where we selected the representatives of each OTU. So we can write OTU underscore to highlight those terminals. So these are the queries in amongst this very large reference tree. And this job is done. So we have placed a set of community DNA of Lepidoptera herbivores from a tree diversity experiment into a large synthesis reference phylogeny, keeping the topology of the references. Now this enables us to have a much more accurate framework for calculation of phylogenetic diversity indices